Okay, now let's talk about the basics of protein structure. So proteins, as we will learn, or maybe as you already know, are big molecules that are just chains of these individual blocks called amino acids. So in the last chapter, we talked all about amino acids, and we started talking about protein structure in terms of the primary structure, which is just the order that the amino acids are linked in, and a little bit about the structure of the peptide bond. But most proteins consist of hundreds of amino acids, and so the actual shape of the protein is what really matters, and that shape is determined by the amino acids. But there are lots of different shapes or conformations that a protein can take, and that is what determines its structure. So, for example, this protein right here is called hemoglobin. So hemoglobin, you may have heard of as an important protein because it is the protein that carries oxygen around in your blood. And it's able to do that because it contains, or it binds to, a molecule called heme, and that heme contains an iron atom, and that iron atom binds to oxygen. So the heme in here, it's kind of hard to see, but it's this little, you can see one here, there's four, so the other two are kind of hidden, but those are the heme groups, and those only fit in there because of the shape of that protein. And then those different subunits fit together because of their shape. In fact, the picture on the right here, this is just the protein, just showing you all the different atoms that make it up. So each atom is a ball, and of course it's hard to make much sense of the shape of this structure when it's in this form. So over here, this is just a different view. Here you're seeing the protein as actually four separate proteins. So we'll talk about that later, but hemoglobin consists of four subunits, and so each of the subunits is colored here. So each one of those is an individual chain of amino acids that are linked by peptide bonds, and then the subunits are linked to each other through non-covalent bonds. There are lots of ways of visualizing proteins, and in fact, since I can just tab over here, I'll show you what I mean by that. So here is this is a web page that where I got these images from, and it's just there are lots of databases like these online that have structural data about proteins. This one has lots of different tricks you can do. So actually, here let's go in here, and just to make it simpler, this is one view of the protein, so I'll change it to appear like it does in the other image. So first of all, if I just come in here, so what I can do from here is I can look at the protein in different views. This is here what the protein might actually look like if you could see it. Now proteins are big, but they're not big enough in most cases to see even with the microscope, although there are some protein structures you can visualize directly, but this is just a mathematical model of what the surface of the protein would look like, and then we can adjust it to show different views to actually show different atoms. So for example, so from this view you can see every individual atom. Carbons are gray, nitrogen's blue, oxygen's red. They don't show the hydrogens, but again, this is just showing you every atom, but we can make a little more sense if we color code them. So here, each atom is color coded by which polypeptide it's part of. So again, this is a protein with four subunits, and so here we can see that again, it's actually four separate molecules, but it's still hard to kind of make sense of what you're looking at. So we can do another step, turn it into kind of a cartoon view, where instead of seeing all the different side chains, you just see most of the backbone. And so these ribbons and strings are parts of the peptide backbone, and the little spirals are what we call alpha helices. We'll talk about what those mean in a minute. But now you can kind of get the sense that these are four different linear strands that are sort of folded up into this complex 3D shape. And again, it's this 3D shape that determines the function of this protein. So like I said, this is a carrier protein for oxygen. So we can even see if I click this. So each of these little structures here is a heme group. So this is one, here's another back here. And then there's two more. So each subunit holds a heme group. At the center of the heme group, this little tiny orange atom here, that is an atom of iron, and so that's where the oxygen would be bound. And again, this property, this ability of this protein to carry that molecule is only possible because of its shape. So that's what this whole chapter is about, is understanding protein shape. So again, we're talking about the structure of proteins, and so there are 4 levels of protein structure that we can talk about. We've already talked about the first level, which is primary structure. So that's what the previous chapter dealt with, and that's just the sequence of amino acids that makes up the protein. Again, this is determined by the gene which encodes the protein. So for every protein, there's a gene, and that gene encodes each amino acid. The ribosome assembles the amino acids in that sequence. We'll talk about how that works later. But then that sequence defines the structure. So there's one end, usually probably down here. This is the amino or N terminus, and then this is the carboxy terminus. Primary structure is always linear, so there's no such thing as a branched polypeptide. And again, if you change that order, if you mutate a gene and change the amino acid sequence of the protein, it will change the primary structure. But then that polypeptide chain coils up or folds up into a 3-dimensional shape, and the first level of that folding we call secondary structure. So the secondary structure of a protein consists of specialized 3-dimensional shapes, which we'll talk about, that are mostly stabilized by interactions between the backbone polypeptide groups. So for example, this structure here, this spiral shape, is known as an alpha helix. So we will talk about how something like an alpha helix can form, and there are other kinds of secondary structures too. And then the next level we call tertiary, which just means like the 3rd degree of structure. And tertiary structure is defined by mostly interactions between the side chains. So again, the secondary structure does not depend as much on the primary structure, but the tertiary structure does, because if you change the interactions between the side groups, you will change the tertiary structure. So it's mostly held together by things like hydrophobic interactions, hydrogen bonding, and so on. And so for some proteins, this is it. So for some proteins, a single polypeptide chain folded up into a tertiary structure like this is the most complex structure it's capable of. But many proteins, possibly most proteins actually form complexes with other polypeptides. So again, a single chain of amino acids is sometimes called a polypeptide, and some proteins only consist of one polypeptide, but many proteins consist of multiple polypeptides, and if that's the case, then we say that protein has quaternary structure. So hemoglobin is a good example. This is just another view of hemoglobin. And so hemoglobin is considered a protein, but it actually consists of 4 separate polypeptide chains that bind to each other and form this quaternary structure. Generally, quaternary structure, actually, if we start at the beginning, primary structure is the most stable. So the primary structure of a protein never changes unless the amino acids at one end or the other get cut off, and that does happen. So that's part of the processing steps of some proteins. If you cut off or cleave the either amino acids from the N terminal or the C terminal, you shorten the protein, and therefore you change the primary structure. But it's very rare to change the internal sequence of amino acids. So you can't chop out amino acids from the middle and connect the ends back together. Now there are changes to individual amino acids, which we'll talk about, where you modify amino acids by adding phosphates or hydroxides or whatever. But generally, primary structure doesn't change. Secondary structure doesn't change very much either for most proteins. So most proteins have a characteristic set of secondary structures that don't change again unless there's a major modification to one of the amino acids. Tertiary structure can change quite a bit. In fact, the ability to change tertiary structure is usually how most proteins function. So the ability to alter their conformation when they bind to a ligand or when they're in part of an enzyme reaction, for example, is how they are able to do that. And quaternary structure also can change. So because the quaternary structure depends on tertiary structure, it can change as subunits bind to each other and release each other. Some quaternary structures are very temporary. So a protein polypeptide might just temporarily bind to another polypeptide as some sort of part of some sort of molecular signaling pathway. And so the actual quaternary structure only exists maybe for a few seconds. Whereas other quaternary structures, like hemoglobin, for example, are quite stable. And in fact, mostly hemoglobin only exists in its quaternary structure form like this. But we'll see, even with hemoglobin, its ability to change shape is a big part of its function. So the forces that hold together the 3D structure of proteins are mostly these weak interactions. So the only covalent interactions within a protein are the peptide bonds themselves, and then these covalent bonds called disulfide bridges. So if you have a cysteine residue, which has that sulfhydride group, it can form a covalent bond with another cysteine through the sulfur groups, and so that's called a disulfide bridge, and that's the only kind of other covalent bond you can have between polypeptides or between sulfurs of the same polypeptide. Everything else, all the other forces that allow a protein to take on these different shapes comes from these weak non-covalent interactions. So hydrogen bonds are really important, especially for secondary structures. So we'll talk about that. Ionic interactions are important if you have charged side chains. So if you have a side chain that has a +1 or -1 charge at neutral pH, then if they're positive, if they're of opposite charges, they'll pull toward each other. If they're the same charge, they'll repel. But probably the most important type of bond or interaction that stabilizes protein are these hydrophobic interactions. So proteins are big molecules with lots of surface area, and water, again, likes to be attracted to other water molecules and to polar groups. So some side chains are polar. The peptide backbone itself is partly polar, and so water molecules like to interact with those parts. But all the non-polar side chains, water does not like to interact with, and so water molecules tend to help force a protein to fold into a shape such that those hydrophobic amino acids are on the inside. So, for example, if we go back to our three-dimensional representation of hemoglobin, you can actually, this little website has the ability to let you look at, for example, the hydrophobicity of your amino acids. So if I visualize that, you can see all the greenish amino acids are the hydrophobic ones, and you can see that for the most part they are tucked deep inside the protein, whereas the more hydrophilic ones, like here's a glutamate, here's an aspartate, here's a lysine, those are all on the outside, whereas deep inside, the dark green ones, for example, tryptophan, leucine, phenylalanine, those green amino acids are all hydrophobic, and the reason they are deeper inside the protein is because they are pushed in there by water. So those hydrophobic interactions are very important for protein structure. Now, but before we start really talking about complex 3D structures of proteins, we have to go back and talk about the peptide bond, because the peptide bond is what holds the amino acids together and determines the primary structure, but it's the shape and the structure of the peptide bond that then leads to the higher levels of structure also. So we know that the peptide bond consists of the connection between the carbonyl carbon of one amino acid and the amino nitrogen of another. So here's the peptide bond of two amino acids. So here's the alpha carbon of one, here's the alpha carbon of another. So there would be, of course, a side chain here and another hydrogen attached to these, but then the peptide bond is what connects them together. And this normally is depicted like this, where you have a carbonyl carbon single bonded to the amino nitrogen, and that nitrogen just has 3 bonds attached to it. But it turns out that this structure, this peptide group, has some resonance structure to it. So the electrons that are shared between the carbonyl carbon and its oxygen atom can kind of go back and forth between the two. So these the electrons that make up this bond can go back to the oxygen and give the oxygen a negative charge, and then the nitrogen, you probably know, has a pair of unbound electrons, and those can then form a double bond with the carbon, with the alpha carbon. So you can have a partial double bond between those two, and then you can also have those go back toward the nitrogen and then get this structure here where it now acts as a single bond, and then the whole thing can go back to its original arrangement. So there's resonance structure here with these 3 arrangements, which means that the actual structure of the bond is some average of these 3, which means that the peptide bond itself, this connection between the carbon and the nitrogen, has a partial double bond structure. And the reason that's important is that if this is a double bond, first of all, this nitrogen will be hybridized in such a way that it'll have a trigonal planar arrangement, just like the carbon would. So in fact, these 4 atoms, the 2 carbons, and the nitrogen, and the oxygen, the carbon, the nitrogen, and the hydrogen, actually kind of all sit in a plane, basically like a rectangle. And that rectangle is basically flat because all those bonds are basically in the same plane. It also means, though, that bond does not rotate. So if we kind of visualize the protein this way, here we've got a chain of 1, 2, 3, 4, 5 amino acids. So you can see the alpha carbons, the R groups, and then the peptide groups as well. Again, that rectangle has been drawn over each peptide group to show that all 4 of those bonds lie in a single plane. So this is one flat plane, and it is defined by the position of all of these bonds. So all those line in a flat plane, and the peptide bond itself, which again is this bond here, cannot rotate or does not rotate very easily. Again, because it basically acts like a double bond, and we know that double bonds do not rotate. So what that means, though, is that all of the rotation of the peptide backbone comes from the bonds between, or that are connected to, the alpha carbon. So for each one, you have each alpha carbon has 2 bonds that make up part of the peptide group, one with its amino nitrogen and one with its carbonyl carbon. And so those are the bonds that rotate because they they are single bonds. And so they rotate, and that rotation is what allows for most of the conformational or changes in shape for a protein. So for each type of bond, we can talk about the rotation angle of that bond. And we use this notation, so the Greek letter phi we use to refer to the rotation of this bond here, the bond between the alpha carbon and the amino nitrogen, and then we use the Greek letter psi to refer to the rotation of the alpha carbon with respect to, or the bond between the alpha carbon and the carbonyl carbon here. So again, we're not talking about the bond angle itself, just the rotation angle of that bond. And this, so if you look at each bond by itself, so this is for example, the psi bond again between the alpha carbon and the carbonyl carbon. So again, what they're not drawing here is there'd be another double bond here with this oxygen, but this connection here, this bond can rotate freely, and so we define it this way. So if you kind of rotate these 4 atoms so that you're looking at them kind of end on, here we have the alpha carbon towards the front and the carbonyl carbon toward the back. And then you can see the 2 nitrogens that are attached to each atom. And that angle psi is defined as the angle that that back group rotates at. So if it's in this position where the nitrogens are essentially opposite to each other, then that's defined as either positive or negative 180 degrees. And then if you rotate it clockwise, so if you rotate it clockwise 60 degrees this way, then that would give you an angle of psi of negative 120 degrees. Another 60 degrees clockwise, and you're negative 60. Another 60 degrees, and you'd be at 0 degrees, which means that the nitrogens are both on the same side of this bond. And that's the furthest you can go in the clockwise direction. Then we rotate keep going clockwise, and we are at positive 60 degrees, and then 120, and then you're back to 180 again. So that's why this position is both plus or minus 180. So each bond has hypothetically that ability to rotate through 180 degrees. In practice, though, not every one of those bond angles is actually feasible, and that's because as you rotate the bond, the groups that are attached to either end, the side chain, which again would be attached to this alpha carbon in this case, so there'd be some R group here, and depending on the R group, what type it is, it's going to have steric interference with either the rest of the peptide group or some part of the side chain of the next amino acid. So that means that some phi and psi angles just are more favorable than others, and which one depends on the amino acid. So in this case, this is called a Ramachandran plot, where basically you plot all the possible phi and psi degrees. So both axes go from negative 180 to positive 180. Again, 0 means this position here, where the angle is rotated such that the nitrogens are opposite to each other, and then 180 is when they're facing at either end. And so again, both angles can go from negative 180 to positive 180, and back to positive 180. And these blue regions represent the rotation angles that are feasible for a particular amino acid. I believe this is for the amino acid alanine. So remember, alanine just has a single methyl group. So the darker the shading, the more favorable that combination of bond angles is. So that means that for alanine, bond angles of, let's say, around 120 degrees, positive 120 degrees for psi, and again that would be that corresponds to this angle right here. So about 10, 11 o'clock in that direction, and then about negative 90 or so in the phi direction. So those two angles for alanine are the most favorable. And then you also see regions like around minus 60 degrees in the psi angle also are feasible. So those are just the most energetically favorable because they allow the least steric hindrance, and then the white areas are generally not allowed, at least for that amino acid. And then plotted on top there, you have little dots from a different protein showing other amino acids. But you may notice some things. First of all, it's not symmetrical, so only that sort of upper left quadrant really has the most favorable angles. That's mostly because these are L amino acids. So if you were to make a protein out of D amino acids, you would see a Ramachandran plot that's basically a mirror image of this. But again, some angles are just not easy to produce, and we'll come back to this later because we'll come back to this in a bit because we'll see that there are some secondary structures that allow different bond angles. All right, so next time we will talk about those secondary structures.